Hey, Patrick.
Hey, Katie.
So, what can we learn from a middle aged, white collar, job working, married father of two children?
Oh, that you absolutely don't need a brain to get yourself through the day.
Welcome back to Unpopular Neuroscience, where we explore popular notions and less popular realities of some of our favorite topics in neuroscience. And we talked last time about insights from simple wiring through the lens of Bradenberg vehicles. And I would like to take us kind of in a very different direction, and that is into the squishy neural substrate that is inside our heads right now, and really take an episode to talk about brains. And I'd like to talk about human brains and animal brains, and probably a little bit about what it means to translate from one to the other in service of sort of asking and answering questions that we find interesting in neuroscience. And if we're going to start with brains, I think maybe a brief tour of how we've thought about brains over time could be a place to start here. And jump in at any time, Patrick, but I think size would be where it was at
Oh
for a
yeah,
very long period, right?
this is a wonderful way to start. I have a lot of experience. My background is mostly I started out as a molecular biologist and worked in neuropharmacology. So I started with very small brains, even sub-brain sized things. And I've kind of worked my way up. The end of my career was very kind of cognitive psychology focused. And so this was definitely an arc that I went through too, of working from the very, very small, very simple uh, you know, electrochemical elements
Mm-hmm.
up to big stuff, complex stuff. So I think this is a great trajectory, and just kind of going through the different sizes of brains is fun and interesting and gives a lot of insight as to how all these different animals kind of go about their days and what their lives are like and what they need a brain for.
Awesome. So size is the place to start. And I think there's size, relative size, and also evolutionary trajectories as well. So, absolute brain size of hominids, it tripled to Homo sapiens over a few million years. There was all these questions of, okay, what do these changes in brain size mean in terms of our cognitive abilities and behavior? And the first thing people want to do always is measure and compare. So this metric called the encephalization quotient, or EQ, was created. And it's something that most people probably have encountered in the popular literature. When they say things have a big brain or a small brain, they're probably talking about this EQ, which is a ratio of brain and body weight relative to kind of a standard expected average mammal brain body ratio. And in this context, one, the standard is actually a cat. So humans, modern humans, have one of the highest EQs of the mammals, from 7.4, 7.5, 7.8. And we often like to see that as a sign of our obviously superior intelligence. I'd say, though, be careful. EQ doesn't have this sort of very nice linear, the higher the EQ, the obviously more intelligent the organism we're talking about. Capuchin monkeys have a greater EQ than chimps or gorillas, but I think if I was a betting person that the chimp or gorilla could outwit the monkey any day on almost any task we would give them.
I think there are a couple of interesting outliers as well. I think squirrels are some
Squirrels and
species of
dogs.
squirrels are really
Yes.
far off the line, higher than humans, and so but we try not to talk about that too much.
We try not to talk about that unless they're really super intelligent, and the food caching is something we can get into later
Right,
as an
right, right,
interesting
right.
behavior.
Then we find a cool behavior, and we're like, no, we were right all along. Right, see, they're obviously very smart.
Similarly, there was also, let's go 19th century, early 20th, let's weigh all the brains. So weighing the brains of eminent people was a bit of a thing. And, obviously there was the ingoing assumption, bigger brains must, really, these people must have been just super cognitively able. And again, the pattern doesn't hold out. Anatole France, his brain, half the weight of Ivan Turgenev, both great authors. Turgenev wrote Fathers and Sons. France won the Nobel Prize for Literature. So, when they started doing these comparisons, it was kind of looking like, well, actually, double the brain size does not have some sort of double the cognitive power, or even not a specific advantage that they could find and point at.
Quick aside, Gould's book, The Mismeasure of Man, actually has a lot of discussion in this area that's actually kind of interesting. It's written sort of as a response to another book, and it's mostly about IQ, which is I actually think not a particularly useful debate to get into, or it doesn't generate a lot of light. But the brain size thing that he talks about, he lays out kind of a historical pattern of look at all these different ways in which phrenology and brain size was used, and look at all the different kinds of measures and all the subtle ways in which you can kind of choose to measure accurately or inaccurately, the examples you want to emphasize and the examples you don't want to emphasize based on kind of your research paradigm at the time. And I think it's a kind of a fun treatment of that, an interesting treatment of that, and very much in response to this period of time you're talking about in the early 20th century, where weighing brains was a big thing.
Fantastic recommendation. And you said phrenology, so let's go there. So it's not just the size, what could it be about? So phrenologists very interested in the lumps and bumps on your skull and how that could relate to elaborations in the structure that's underneath, and maybe that links to personality traits or abilities. Thoroughly debunked, but it was another big craze. If it's about the structure, maybe the folds started to get people's interest there. So we talk about cortical folding a lot. Then more conversations came into looking at composition. So people have probably heard about ratios of white versus gray matter, the types of actual neurons, so double bouquet cells and things like that. If you're in my neuroscience class, we kind of get into looking at histology and slices of different neurons and looking at the structure that they have. And then into other questions around organizations. They love to take slices of the cortex and look at the layered structure there and say, ooh, look, human, this many layers, cat, this many layers, rat, this many layers, organized in this way. And then that leads to further questions about anatomy versus function. And I think you actually have a paper about this, Patrick, if I'm not mistaken.
Yes, my least popular paper, comfortably least popular paper, is actually on this very topic. I'll talk about this, I'm sure, in some other episode. I was part of a large cognitive training study to try to get people to see if we can, through intervention, change cognitive abilities, right? And so we had this wonderful data set with literally thousands of people who had all kinds of interventions. We did electricity through their brains, we had them play video games, we had them meditate, we had them do physical fitness, and we had them take tons of cognitive tests. And we got to see all this longitudinal data over a 6 to 9 month period. It was a long study, and thousands of people. It was a huge deal, very expensive to run, and with not spectacular results. Basically, we didn't see a lot of change in any cognitive abilities. They learned to do better at the specific tasks. And we could show some modulation of the different treatments that made them better at those specific tasks. We didn't see a bunch of big cognitive changes. But one of the major things that you have the ability to do is to look at neuroanatomy across these. I had hundreds or thousands of anatomical scans. And I could ask questions, which are very often the way that these questions are asked is they say, who has the bigger brain? But there's another way you can turn that question around, and that's what's in this paper, which is, how do brains vary? Right? Yeah. Is size the most important thing that brains vary on? Right? And remember, I have cognitive stuff in there. I have all kinds of factors that I could look at that could influence size and shape of brain. And I did a kind of exploratory factor analysis where I said, okay, what are the major factors that vary across brains? And I threw in all kinds of, I use some nonlinear PCA techniques so that you can throw in categorical variables as well. So you can look at gender, you can look at people's scores in these cognitive tests. So we had personality tests as well. And then all these anatomical variables that are measured with MRI. What comes out, I thought was very interesting. I read a whole paper about it, but apparently I'm the only one. No one else has read this paper. And the upshot of it is that there is the first largest factor, which is something we usually do control for, is brain size, right? It's just kind of how big your brain is. And this is very, very tightly related to body size. People with big heads tend to have bigger brains. There's just more space in there, right? If you've got a big body, you tend to have more brain volume. Volume, I don't know about cells.
Volume, yes.
Yeah. But that's not the only consequence. And this is something that I think is really, I thought this was really interesting. And I will do my best to sell it here. So one of the things you normally do when you're looking at different anatomical volumes in a brain, suppose you want to make a claim that, okay, this treatment helps increase the size of your hippocampus. You'll see papers sometimes like this with older folks. It's like, go out and walk and it'll preserve your memory because it'll increase the size of your hippocampus.
Exactly. Cognitive those protective factors, keep dementia away, use it or lose it, those kinds of things, right?
Okay. So one of the things that you'll find is, hey, if you have a bigger head, you'll have a bigger hippocampus. So maybe people who walk further also have longer legs.
Of course. And they would. Yes.
And they would.
Yeah.
And the important element to that is you can't fully control for that, or it's harder to control for that than you think. Because
Mm-hmm.
even though you can include that as a factor in your model, if that's the largest single factor in your study and you make a mistake in your control, well, then because it's the largest factor, it's going to absolutely overween all of the other factors that you're looking at. If there was a tiny change in the volume of their hippocampus, but you didn't
Mm-hmm.
adequately control for the length of their legs, that might actually mean that all of the effects you're seeing are just due to leg length, because body size is such a huge factor relative to the little tiny, a couple milliliter variations you're going to
Mm-hmm.
have in that hippocampus. And so it actually really matters to get these things carefully measured. And it turns out that there are those big factors have downstream consequences. So the biggest factor is like head size, and that typically
Yep.
drives a reasonably noticeable gender difference in
Mm-hmm.
volumes. So people who mark male on the form tend to have larger heads than people who mark female on the form,
Yep.
but
Mm-hmm.
they kind of have the same amount of cells. And
Yep.
so what that means is
Mm-hmm.
people who mark female on the forms will tend to have more gray matter because it's a little more dense, it's a little more packed in,
Yes.
right? Now that's an
Interesting.
interesting difference, but it's
Yeah.
straightforwardly downstream of just head size. We talk a lot about folds on the brain being
Yes,
important.
absolutely.
Now that
Yep.
actually is we're going to talk about animal brains here in a minute. That is
Mm-hmm.
actually sort of a human accident, which is a
Yeah.
consequence of us having to pass our brain through a pelvis, our remarkably
Yes,
large
it
brain through a
is.
pelvis. Most animals have relatively spherical-like brains, and that's because they aren't trying to grow a huge brain in a compact space. If you try to grow this
Mm-hmm.
huge brain in a compact space, you get all of these sulci and gyri,
Yeah.
which are the bumps and the lumps on brains, because you kind of cramming, like imagine you had a,
Gotta fold it into a
yeah,
smaller
imagine you
spot so
had
you
like
can
a
wrinkle
napkin,
it
you're
out.
cramming it into this small space, it
Yeah,
would get all crumpled
exactly.
up, right? That's what's going on. If you have a bigger head, therefore, your napkin won't be
Mm-hmm.
quite as crumpled up. Another anatomical difference that I was able to observe is that there tends to be some major variation in how big your frontal cortex is versus your parietal and
Mm-hmm.
occipital cortices in the back. And
Yeah.
I don't know for sure, but I bet that that's a neck angle phenomenon.
Ah, interesting.
Humans are a
Yeah.
weird
Yeah.
species because we have our,
We
our
are weird. Yeah,
our head is bent down.
absolutely.
Most animals, if you look at the way that their brain is laid out, they have kind of a couple little olfactory, most mammals have a couple little olfactory bubbles off the front. Then they have this sort of
Yep.
heart-shaped cortex behind that.
Yep.
And then their cerebellum is a little, it actually does look like a bell in animals. It means a little
Yeah.
bumper. And it's kind of snuggled in there behind. And then their notochord,
Back. Yeah.
their spinal cord goes straight back. And humans have rotated this forward, right?
Mm-hmm.
And there are consequences to that in how your brain forms, because now your brain is forming out kind of forward and backward as opposed to
Mm-hmm.
just growing on top and forward. And
Yes.
so you'll get variation in size, and it's probably related to that neck angle. The reason I bring all these up is nearly none of these correlated with the cognitive factors that we have,
Yeah.
right?
Yeah.
Except in sort of ways that are much easier to explain with things like, oh, this person is 60, and therefore their brain
Mm-hmm.
volume has decreased a little bit because they're older,
Yeah.
and your brain volume has
Yeah.
decreased over time. And they scored a little bit lower on some of the reaction time tests, which is very common, right?
Very common. Yeah, expected.
And so so I like to point to those and say, like, there are these gross factors in anatomy that are very difficult to control for, but seem not to matter that much, right? When you actually look at the cognitive factors that are going on. And when they do seem to matter, there's almost always on the ground a much easier explanation that is very concrete, like aging or head size or things like that.
Yeah.
And if someone is ever giving you the hard sell that this part of the brain matters a lot, or that there's a big gender difference in this sort of thing, right? I immediately get really suspicious because I've done the anatomy. And the thing that I learned from the anatomy is the big factors are things like neck angle and like skull size and things like that, and these weird like head geometry sort of thing. And they all seemed very developmental. And none of them seemed to actually be cognitive in any meaningful way. And no one can really come at me with data after this because I had 2,000 samples. And so people will come up with like 16, they'll scan like 16 people and they'll be like, see, we found a factor. And I'm
Well,
like,
see,
go
you see.
away.
No.
So that was my
it's-
experience. That's my least popular paper. And it seems
It's a
to,
good paper.
Yeah.
I'm sad that it was your least popular because that's a lot of scans that you've
was-
done and a lot of factors that you've investigated. That's a
was a
lot
very
of
expensive
work. I will.
paper.
That's a very expensive paper. So this is interesting because I said we want to talk about brains and brains that we find interesting for various reasons and the kinds of questions they can help us ask and answer about cognition, consciousness, intelligence, behavior, all of that. So if humans are that messy and it's hard to find things that matter, what's the point of comparative anatomy? Because I think people do work in non-human subjects all the time, and then the goal is to make inferences or develop our understanding in ways that can be consequential to our understanding of human cognition, behavior, neurodegenerative diseases, things like that.
Yeah, this is actually, I wanted to take a second here for us to talk about our different experiences, because coming from a
Mm-hmm.
computational neuroscience background, the funny thing for me is like we'll do comparative anatomy stuff. We'll be
Yeah.
modeling like cockroach tripod walking gait is a good example
Mm-hmm.
here. And the computational neuroscientists will be like, and that's how walking works, right? And they don't
Yes.
mean for a cockroach. They mean like, well, we've got walking work down.
Yeah.
And it's in part because they don't have deep knowledge about like other brains and other systems. So they
Mm-hmm.
because they don't have that knowledge, they almost can't make the critique that like
Mm-hmm.
this transspecies cognitive compare or these transspecies brain comparisons are not a great idea, or are more complex or hard to know. But you, I think,
Yeah.
have a very different experience where this is a critique that comes up all the time where people say like, well, I can ignore the cockroach walking behavior. There's nothing there for me.
Yeah, and I think this is where all of the canonical animal models, being all the way down to Drosophila and C. elegans, are where you're saying, okay, we're going to try something typically genetic, I guess, and then try to look at homologous genes and make inferences about what that means for humans. And I'll talk about one of my favorites there shortly. But certainly, the rodent models, I think, is where a lot of neuroscience happens. I think there's millions of subjects every year. It's millions in just individual countries at this point.
Yep.
And that is where there's lines to which you look to make comparisons. So I think the genetic piece more and more is at the forefront. But certainly there's these accepted mappings, I would call them. Conferences, you go to SFN and you walk into a room where there's the rat people and maybe they're interested in cognition, in Parkinson's or Alzheimer's or schizophrenia or something like that. And they'll say things like, well, we did this intervention targeting the ventromedial frontal cortex of the rat, which of course is the analog of the human dorsolateral frontal cortex. And everyone earnestly nods along. And then there's questions they'll ask, but they're not going to question necessarily that mapping. It's kind of an accepted thing in the field. And then at the same time, I think the popular perspective on that goes in two directions. I think one direction kind of nods along to authority and says, like, well, they
Yeah.
obviously figured this out. And so it must be this very direct, obvious, not just surface validity, but like deeply valid. And then the other group says, how on earth can you possibly make a statement like that? Human behavior, particularly the kinds of executive function that
Yeah.
associated with the dorsolateral frontal cortex, that's not the same thing. And that's, we can get into the ethics and practicalities of animal experimentation very quickly if we go down that rabbit hole. So I'd suggest we park it for another time. But I'd say out there you will encounter both of those perspectives. And to my mind, the truth, or what I think of as kind of the more unpopular, caveated, nuanced perspective, is somewhere in the middle, where I'd say there's utility in that mapping, there's reasons for it, and then at the same time, I think we want to get into questions of, well, what is this behavior that we're observing in the animals, and how are we describing it in human terms, particularly for certain, maybe what we call higher order cognitive functions. But I hesitate to use that term
Yeah.
because it already sounds pretty values laden.
It makes a little contact with some of the stuff I think we talked about in our first episode about when you're investigating something scientifically, you view it as this very kind of smooth, continuous, complex, nuanced space. And
Yep.
you're just trying to get evidence in there. And
Yep.
so if you have some evidence that rats behave in this way and that it's related to this particular brain structure and that there's some influence over it,
Mm-hmm.
as a scientist, you're like, oh, great, okay, I know a little bit more now. And I think that that's very different from a lot of the way that kind of popular knowledge works, because popular knowledge is very fact-based, and it's more
Yeah.
about being able to either, in the authority case, to respond in the way the authority wants you to, right? Very familiar for students, of course.
Mm-hmm.
Or just to kind of make sure that you're all kind of on the same page, like to have a coherent, shared understanding of things.
Yeah.
And in the sciences, we just don't care. We're not trying to have a shared understanding of things. We're trying to like accumulate evidence. And the judgment is actually something we almost don't do. The popular neuroscience science often says, okay, taking
Yeah.
this evidence, here's the synthesis. And scientists, we don't do the synthesis. It's not part of our job description. We don't care, right? We're not engineers. It's not our problem, right? What's this for? That's not my. I don't know. You go ask an engineer. Go ask a doctor. I just need to find out. And so I think that a lot of this comparative anatomy falls in that category.
And there's something you said earlier, which I think can also be reflected in sort of the popular dichotomy that I described, which kind of implied that a question along the lines of how can you make these comparisons if you don't fully understand everything about the animal in question. So I'm going to give us a challenge, and this could backfire spectacularly, but let's try anyway. I'm going to put a question to us, the comparative anatomy question. I'm pretty sure neither of us know the answer to this, so speak up if you're going to be cheating. And then I'll say let's do like the 30-second Google, because I think that in general, there's enough similarities that if you have a few points of reference to go on, most neuroscientists would be willing to kind of place a bet on what they would expect to find in a brain. So songbirds, let's think about them.
Okay.
Human language, we often talk about it as being lateralized or left lateralized. Yep. So we see more elaboration on the left side of the brain for that.
A little more complex than that, but yes, give you some rope here.
You're welcome to. Do you want to do the 5-minute explanation?
No. I do the bet first. I want to know the bet first. I enjoy betting games.
So here's your betting game.
Okay.
Do we observe left side lateralization in songbirds that would appear to be connected to the nature of their sort of what we call linguistic ability, their singing?
Okay, I like this question. And I'm going to try and think aloud through it. I'm going to try and say the things that I know.
Do.
Okay. So first, I know that birds have a different pattern of development. They don't have a cortex. The neocortex is mammalian. Yeah. Mm-hmm. But they have a sort of an equivalent. They have a developed forebrain.
They do.
That fulfills a similar role to cortex. Mm-hmm. So we're talking about the stuff on top, but it's kind of more, maybe it's more in front on birds than it is on top, right? Mammals have this, neocortex means bark, new bark, and it's evolutionarily new. That's the neo part, and cortex is bark. So it's this bark over their head, and that's the wrinkly stuff. And birds don't have that, but they do have kind of a big ball of forebrain material.
The big ball, the pallium, I think it's
called. Pallium, okay, yeah. I'd forgotten the word. Okay, so far so good. So it's going to be in the pallium, is my first guess, and not actually in the brain as a whole, because it's going to be a little bit different. So if we're making a bet, now I'm thinking about a subregion of the bird's brain, but it still has.
You're making it even more specific. Excellent.
Yeah, see, it's now lateralized, but it's still lateralized, right? There's still a left and a right, and it still develops in this. There's still a central fissure. Now we have to go into the part about language lateralization. I have to think a little bit more about this, because while it is popularly said that language is left lateralized, that's only mostly true. We're asking the word language to do a lot of work there. Because what is usually going on, what people usually mean is grammar or sort of the generative rules.
Yeah. So you think of Broca's aphasia, Wernicke's aphasia, people probably have heard of Broca's area and Wernicke's area, and these are the kinds of structures and processes we're talking about here when we're saying left lateralization.
Yes, a Broca's aphasia is kind of near the motor cortex. And it's a breakdown. It's if you have a lesion near the motor cortex, you may lose the ability to speak fluently. You can still get words out, but it takes you a few seconds to generate each word. It's like you're snapping them together individually, rather than having this lovely fluent chain of language generated for you. It's a classic agrammatic aphasia. And Wernicke's aphasia, you can generate those sentences. It's a conduction aphasia. It's a white matter lesion, but you can generate those lovely sentences, but they're wacky and meaningless and only like vaguely, like you kind of got the wrong words snapped in there.
It's word salad.
It's word salad. Both of those are left lateralized.
Sounds fluent.
However, there are right lateralized effects on language, and the right lateralized effects on language are, interestingly, more pronounced in things like singing and music.
So here we go.
So the knowing emotional tone tends to be a right lateralized function. So if you only have someone's body language and tone of voice, if they're speaking another language, for instance, and you're trying to say, like, what are they saying? What are they feeling? What are they thinking? If you have right lateralized lesions, it's actually more difficult for you than if you have left lateralized lesions. So that's really, like, so now I'm confused about songbirds. So is it left lateralized? It's like, well, okay, here's my bet. Here's the bet that I would end up making. I bet that generating kind of like fluent
Mm-hmm.
songs. So like note
Yep.
transitions, fluent changes. I bet that's left lateralized.
Mm-hmm.
But I bet that there's something like ability to sing to females or something like that, like so using them socially or something that you
Mm-hmm.
could also find right lateralized. And if the whole thing
Mm-hmm.
turned out to be right lateralized and it's just like, nah, it's just where music is, I would buy it. Right. So like there's a
Yeah.
range of acceptable answers, but
Mm-hmm.
that's my bet. Okay, there's my bet. I say I bet it's left lateralized in terms of like fluency, but if it turns out to be right lateralized and it's like a music related explanation, I would say like, yeah, okay, you got me on that one. I'm comfortable losing the bet that way.
Like you've talked this through in far more detail than I had because I was picturing being in a bar,
Oh, okay,
slapping
okay.
a fiver down on the table. But similar lines of reasoning. I think looking at the humans, trying to think about what we think is going on in bird song and the learning and memory and the way they assemble and the sort of parallels people sometimes make thinking about grammars and that. So my Googling, this is where I think people will need to go off and do their own
Right.
research to look at the nuances that you're getting into. But I am seeing Nature paper, hemispheric dominance underlying the neural substrate. Songbirds, the avian higher auditory cortex is left lateralized for processing song memories in juvenile zebra finches. There's evidence for lateralization of neural responsiveness in brain regions. And this is another paper about humans and
Yep.
songbirds looking at infants, language development. Looks like, yep, in awake birds, they're seeing some differences. So
Yep. I
there we go. We
and
can
and
reason our way through some of these questions
and talking about
about sort of
memory, I think
comparisons
that's
across species.
sorry. And talking about memory, I think that's exactly the area that I was trying to like make that differentiation. I was like, if you're talking about like something more systematic or something like learning
Mm-hmm.
something, yeah, I bet left lateralization is a reasonable bet. So, do you want me to
All
make one?
right, sure, if you want to.
Okay. One of the rarest animals in the world is the okapi.
Okay,
Right. It is
tell me
a
more.
forest dwelling ungulate. It's a quadruped.
Mm-hmm.
It's sort of like
Okay.
a zebra. It's got stripes. They're
Yep.
so rare that you just occasionally see them on game cameras. They're almost never. We've
Okay.
never captured one. I'm sure
Mm-hmm.
comparative neuroanatomy has never been done on them. But
Mm-hmm.
I bet just knowing the shape of the okapi's head, which is deer-like,
Mm-hmm.
you can
Yep.
tell me whether they have ocular dominance columns like a cat
Mm-hmm.
or like a
Yep.
rat.
Ah, rat. Rat. I guess rat. And you're nodding, so I'm assuming we would bet in the same direction. So
Gotta be
my
right.
reasoning here is ocular dominance columns are a feature of mammals like humans in particular that have binocular vision. So our eyes are on the front of our head and we're using it for depth perception and guiding a lot of behavior. Rats or animals that don't have ocular dominance columns are certainly not ones that are sort of as clear and as well structured in the visual cortex as humans and cats. Eyes are more on the side of the head, so they're not using binocular vision. They're going for field of view. They're typically prey animals. Something like a copy, they're out there, and what are they doing? Well, they're probably outwitting blades of grass and trying not to be eaten. So they're not going to be relying on binocular vision for hunting and creeping around and engaging with their environment in that kind of a way. So I would be very confident that although, as you say, the answer to this is probably unknown, that we would be betting in the right direction on this one.
Yeah, I agree with that. And if you are like a rat or a prey species, they're pretty much just in their higgledy piggledy, right?
Yep.
If you have binocular vision, if you're a predator species, just like
Mm-hmm.
we talked about in our vehicles episode, you have these
Mm-hmm.
arrays of cells, and you need to do a comparison between right and left eye. And so you have these columns where you have a layer of cells that respond to both eyes, a layer of cells that respond to left eye, binocular cells, right eye cells,
Mm-hmm.
binocular cells, left eye cells. So these nice columns so that you can make those comparisons between the two different eyes. Okapi's got to be a prey species, very shy. They ain't out hunting. I'm betting higgledy piggledy like a rat.
Let's get into some brains or animals that we think are interesting. And I would say just think of your favorites and maybe we can just take turns highlighting what we find interesting and ask some questions of each other if we're interested in doing that at any point.
Awesome.
So I would start where I got started, which is with C. elegans. These are the little, very small roundworms. They are a model organism, and with great thanks to Dr. Kathy Rankin, who took me into her C. elegans lab when I was an undergraduate student, I got to play around with learning and memory in our little C. elegans. And what is so cool about them is they are almost like the vehicle's perspective. We can really understand the wiring diagram that's there. So they have 302 anatomically mapped neurons. It's a complete connectome. Like I can show you the wiring diagram of this organism. The genome's relatively simple, so we've got a pretty good understanding of what the genes are, what they do. We've got conserved genetic pathways. So what that means is you can look at the genes and signaling involved in certain neural functions, and it's going to be, I'll put heavy quotes, identical or at least very similar between C. elegans and more complex organisms. So we have this idea of canonical homologous genes, back to my mapping of brain regions kind of ideas from before. They're nice and transparent, so you can image them as things are going on in them. And more recently, all these neuropeptide interactions actually got mapped out and understood. And there's, I think, about 30,000 of them. So this is a pretty small thing. They have a short lifespan. I think it's a couple weeks. So you breed them, experiment with them, and then you can do it. High throughput is a term we use when you can just have a large number of subjects run very quickly, often cheaply is the other connotation there. So this is not your 2,000 people in a scanner.
Yeah, and they're about
Months.
they're visible to the naked eye, but only
Yeah,
just.
like about a millimeter. They're just
Yeah,
barely there. You can look on your plate of agar and say, like, I see you. They can sort of see that there's some
yeah.
movement, going on. But then you just bring in a light microscope and you can see very, very effectively. So, what questions do we explore with this organism? And I find that really wild because I remember saying these are my drunk tapping memory
Yeah.
habituation thing I talked about in our first episode. We were asking questions about the neurobiological basis with which alcohol affects memory. And that sounds really wild, but you say, look, we've got developmental trajectories that are so short, you can expose them to ethanol and then see not just the impact on the worms that initially develop, but several generations later. So those super interesting developmental questions you can get at there and look at epigenetics as well. Behavior, they have basic sensory processing. They have what we call learning and memory, but we can dig into that with aplesia, which I'd like to talk
Mm-hmm.
about later on. Aging, synaptic function, plasticity, all of these are things that can be explored. And in fact, there was a Nobel Prize went to C. elegans research where they were looking at the genetic basis of apoptosis or programmed cell death. And this was a piece of work that they really felt, due to the homologous genes, they could readily translate interesting findings all the way from these almost microscopic worms all the way up to people.
Yeah, I am actually reasonably familiar. They're probably the most complex face-up neuroscience model.
Mm-hmm.
Like the nervous system that they have, we know the explicit wiring diagram. We
Yep.
know exactly how the cells differentiate, and they are wired up sort of precisely. Like
Mm-hmm.
each cell makes specific contact with specific other cells in a way that is kind of
Yes.
nameable and describable.
Yeah.
And
Yes.
there was a big story recently about Europe trying to like simulate an entire rat brain or an entire rat
Yeah.
cortical column. And one of the things that is true about that is those are randomly positioned and randomly connected cells.
Mm-hmm.
There is a random dice rolling model that lays
Yeah.
out that thing, and that is not true in C. elegans. In C. elegans,
Mm-hmm.
it is precisely known. Is there a precise? Could we have precise knowledge of more complex nervous systems? Maybe not.
Maybe not. Yeah, no,
Right?
I'm with you on maybe not.
Yeah,
It's
they
much.
might be randomly rolled, but it's really nice to have a model that you actually know what is supposed to be connected up to something else. If you have a question like how does alcohol affect this system, you can know.
Mm-hmm.
And in a rat, you might be like, well, either the alcohol affected the system or that was just wired that way for random reasons that are pretty common, and we just don't know. So there are reasons to use such a simple and face up neuroanatomical system that I think are important, and it would be natural to use a system like this one.
Agreed. And it's one where I'd encourage people just to go away and have a look because this is all over the internet. Like you can go up and there is the database of all of the genes that are understood in this worm. And there's the full connectome modeled online. You can see the circuits that we're talking about
Yeah.
here. And there's some great work they've done with fluorescent proteins as well, which are pretty cool if you'd like to see some glow in the dark worms kind of worming their way around and doing things.
It's one of the big model organisms. I generally judge how big a model organism is by how likely it is for me to know their Latin name. And they're only about 10 where I'm like, I know the Latin name because I know it from research. And there are only a handful like that.
Awesome. So how about you? What's a sort of smaller scale organism of interest?
Oh, one of my hands down favorites. And this is one that we learned. I actually, when I was in Bill Brewer's kind of cognitive neuroscience class,
Hmm.
he did a bunch of comparative stuff where he said, like, let's ask these questions of simpler nervous systems. One of the ones
Hmm.
we started with fairly early on is the Sulcidae, the jumping spiders.
Oh, yes.
And they are bananas. So they
Hmm.
are a very tiny spider. They're about 4 millimeters square, something like that. They're about the size
Hmm.
of a cap of a pen is maybe a good
Okay.
good
Yeah.
comparison. They have a tiny brain. It's
Hmm.
order of 10 to the fifth cells, that sort of thing. 10 to the fourth, 10 to the fifth
Yeah.
cells. The Like most invertebrates, they have, or like most arthropods, it doesn't look like a mammal brain at all. It has these like mushroom body things. They kind of
Oh, okay. Yeah, yeah, yeah.
they kind of look like a motorcycle panhead engine, if you know what one of those looks like. It's like a cross section of that. And they are so you wouldn't expect them to be particularly bright,
Hmm.
but as far as we know, they are. There's this wonderful paper that I love to cite about people do called eight-legged cats,
Okay.
because their ability to solve a lot of kind of cognitive science problems is cat-like. Like they are
Interesting.
that smart.
So give me an example. Like, what's an example of a problem-solving jumping spider?
A couple great examples. So one great example. So first of all, jumping spiders they hunt other spiders. Their prey species is other
Oh,
spiders, right? Which might be
okay.
part of why
Yes.
they need to be fairly cagey, right? They don't spin webs. They are active hunters like wolf spiders or something like that. So one of the ways that they are tested is you take a spider, a prey species spider, and you spray it with shellac so they can't smell it,
Hmm. Hmm.
and you put it on top of a 3D wire maze. So you have two little wires that it could climb, and you
Hmm.
take them and you twist them all around each other to create this really complex geometric shape like a tree. And you put the spider at the end of one of them,
Hmm.
the prey spider. You put the jumping spider down, and they have exquisite vision. So
Okay.
they have 8 eyes. They have 6 small eyes, which kind of have light and dark, or kind of like a fly's eye
Hmm.
or something like that. That give them all around vision. And then they have these two beautiful like tubiform eyes on the front.
Oh, interesting. Yeah.
So they don't have lenses like a human eye where you kind of smush the lens
Hmm.
back and forth to change your focus. They actually have these long tubes, and they can flex the whole tube and like turn it around at different angles so that they can see in different directions and focus on things like in this
Hmm.
huge field of vision in front of them, but they have tight focus on it. And so they'll sit there and they'll look up at this tree for
Hmm.
20 or 30 seconds. And then 90% of the time they'll climb the right tree. Like they'll work it out.
Okay.
They'll look at
Yes.
the structure and say like, okay, there's where I want to go. There's the spider.
Hmm.
Okay, so I need
Yeah.
to make a left turn,
Hmm.
then trace it back down. Okay, it's this one, and then they climb up the right
Yeah.
one. They get the spider almost every time. So they're working out somehow,
Hmm.
like
Yeah.
what the bends of this thing are. Right, that's pretty complex and shows, I think, some degree of internal representation beyond what we would expect of the Breitenberg machines. There might be like there's probably some external loading of that where they're looking at the tree,
Hmm.
but that's a pretty complex idea that they're working their way through. And
That's
that's
a
if the
very
spider,
complex idea.
yeah,
Yeah. And
and
I like the fact that there's what I'd call initial thinking time. If I use a term from the human literature
yeah,
on, say, like
yeah,
Power of London task and things like that, it's the Bradenburg vehicle would start down one of the paths and probably bump its way along, and
right,
would be figuring it out. And even that's a very
50-50,
generous
right,
term, as it went.
yeah,
But to have something that observes, as you say, some crunching's going on, and then the accurate goal-directed behavior takes place. That's really interesting to observe that in something on the scale that we're talking
yeah.
about here.
And that's a fairly, what you might call ecologically invalid task. It has some ecological validity, but in the wild, they are unbelievably fiendish. So I said they hunt other spiders. They will go
Yeah.
up to other spiders' webs.
Hmm.
They
Hmm.
will put a finger on that web, or an arm on that web, and depending on the species of the spider,
Whoa.
for certain spiders,
Yeah.
they will wiggle as if they are prey caught in the web to lure
Hmm.
those spiders out. For spiders that are not responsive to that, or that tend to approach prey trapped in the web aggressively,
Hmm.
instead they will wiggle those webs as if they are a male of that species who has come to mate.
That is wild.
They have
That
lots
is
of tricks and
wild.
traps. They are very clever. They're such clever, and this 1,000, 10 to the fifth, tens of thousands of
Hmm.
of
Hmm.
neurons. doing this computation. Really interesting, really, and I think part of why we regard them as intelligent is that they are such visual creatures like us, and they engage with their
Yes.
world in that
Yeah.
way.
Yeah.
But all really fun, and they're really cute. There's a species of them that does these elaborate dances.
Oh, okay. This needs to be. We got to look for video.
Yeah. I'll include peacock spider dances. Yeah. So to
Yes.
attract the attention of their own species, they have these
Hmm.
hilarious little dances where they put their legs up and they have beautiful like peacock like beautiful colored tails because their vision again is so good
Okay.
that they have these
Yeah.
beautiful colorations and patterns. So I love them. Jumping spiders are the best. Best spider. 10 out of 10 spider.
Best spider. All right. So I'll take us up in like growth scale, but not necessarily number of neurons, because I'm going to go with something that's bigger, but probably fewer neurons than you're describing here. So keep on with the Latin names. Aplysia, or
Yeah.
the humble sea slug, is probably more what people would think about. So bigger than a jumping spider, but I think maybe only around 10,000 neurons in their sort of ganglion system there. This is another Nobel Prize organism. So Eric Kandel used these sea slugs to really try to understand the biological basis of short-term and long-term memory, and really got at that interesting distinction between changes in connectivity or the likelihood of activations from one synapse to another versus changes in anatomy, such as new synapses or new protein synthesis taking place. And this is one of those creatures where I am an ocean person, so I find them really fun to go and discover when you're out free
The
diving and sort of swim around. But
I was gonna say the
simple nervous
scam.
system.
If someone is studying like octopi or lobsters or
Oh
sea
yeah,
slugs, you know that they came from like a landlocked Midwest university and they picked that organism so they could go diving on the Florida Keys.
straight to the coast, straight to the coast. Because of course you need to gather your specimens and maybe observe them, in
Just
their
doesn't work
natural. And
otherwise.
just it just doesn't work otherwise. But sort of similar to the kinds of things that made C. elegans of interest to us. We have a simple nervous system. They got large neurons, so you can observe or understand where they are, what their function could be, and what I'd call basic reproducible behavior. So there's some responses that they have that you can consistently see across all of your subjects. In the C. elegans, when I was doing the getting them drunk and do they learn and remember, I was using something called the tap withdrawal response. So it's a habituation to a stimulus of just actually tapping their petri dish that they were in. And this is one where there's two things to highlight here. I think one, again, more simple organism, simple nervous system, really interesting insights. But also, let's pause for the popular perspective here, because I've been using terms like memory and
Mm-hmm.
learning and all of that, and I'm sure people have seen news articles or headlines of like, oh, the slime mold that shares memory by fusing was one we read in my class, or flatworms, they eat each other and transmit memory. What's your reaction to that? Because I think you have some strong opinions about clickbait
Oh
here.
yeah, I mean I think it's obvious clickbait. I actually know this for planarians, which are another flatworm. You can grind up planarians, feed them to other planarians, and then the new planarians will do the things that you taught the old planarians to do. A classic example is like tap withdrawal. You condition planarians to bunch up when you tap, and then you grind them up, you feed them to other planarians, and then the new planarians will also bunch up when you tap. It's like, wow, they transferred the memory. Now, what they are trying to get you to believe that it's working like in the Matrix or something,
Mm-hmm.
where you have this like explicit experiential response of that memory, and now you are having that explicit experiential response of that memory as a different or in a different organism. It's like remembering a previous life or something like that. Is absolutely not what's going on. It is like if a better example would be if
Mm-hmm.
if I came up to you and I hit you with a stick over and over again until you were very sore all
Yeah,
over,
very
right?
sore. Yeah.
And anytime you'd be touched, you'd be like, ouch, right? And you just kind of
Yep.
flex. And then I took out all of your cortisol, like your stress hormones, and injected them into
Mm-hmm.
somebody else. And now that person would be like, my skin's a little more tender than usual,
Yeah, I'm feeling a little
right?
jumpy.
That's
I'm
actually
gonna
what's happening.
respond.
That's
That's
actually what's
exactly what's
happening.
happening.
And so it's a clickbait because they're trying to make you believe that it's like the Matrix, and it's
Yeah.
really more like someone just stressed you out, right? We took a stressed out person, we took out
Yeah.
the thing
Gave
that
the
makes them
stress
stressed out,
juice to somebody else,
and
and
gave
oh,
the
that
stress hormone
person's
to
out
someone
stressed
else, and
out.
now
Yeah.
now they seem a little more stressed, right?
Okay.
Oh, and the magnitude tends to be small, too.
Yeah.
I was actually on a thing where this was being done kind of in rats, and they were doing it in a very like sophisticated way where they were actually trying to do the Matrix thing. Like they had
Okay.
implants and they were actually playing back patterns of
Oh,
firing,
yes. Okay.
right?
Yeah.
And they were trying to make the argument that they were transferring this memory. But one of the ways that you could know that that's not what's really going on
Hmm.
is that when you trained the original rat and that you
Yeah.
recorded from, they were getting the task right like 99% of the time. Like they were trained to criterion. So they basically never
Yes.
fail.
Yeah.
And the new rat was just barely beating chance.
Oh, there we go.
Right.
Yeah. Yeah. Yeah.
And so if it's having the memory, why is it so bad
Yeah. This
at the
is
task?
something else, as
It's
you say.
interesting.
Yeah.
It's interesting that it works,
It's
but
very
it
interesting.
ain't the Matrix. It ain't the Matrix. But they
Yeah.
really want you to believe it is. They really would rather you not think about it. It's your turn. What are another?
Okay.
Yeah,
I am a big fan of corvids, and I feel like we've upped the ante into the what is it like to be kinds of questions. So maybe we'll get into some behaviors that we
yeah,
just look at across species and go, wow, that's pretty interesting stuff. So when I say corvids, crows, ravens, rooks, magpies, jackdaws, jays, they're all over the world. There'll
yep,
be some local analog of the ones that I'm most familiar with. And when we think of them, we really highlight some behaviors that look particularly quote unquote intelligent. So, food caching. They hide food for later. They have incredibly accurate spatial memory. So, even 9 months later, that bird knows where it put that acorn or that seed or piece of food. Scrub jays, certain species, they cache perishable and non-perishable foods, and their behavior suggests they kind of know the difference. So, after a certain period of time, they're not going back to check on that perishable food cache because it's not going to be good anymore. So this feels like they have an elaborate sense of what, where, and when, which is kind of maybe starting to feel more like that episodic idea of memory that you talked about before. And that's a piece that people point at. I don't know if you've seen this in person, but I saw this spring, theft and hiding and the sense of social cognition. So I watched one of my local crows. It had a little treat and it hid it in the gutter of a neighbor's garage roof. And then it kind of walked away a little bit, but it didn't leave. And then I noticed this other crow that had been observing flew away. And crow number one went straight back, grabbed the thing that he had, ran a little ways
yeah,
away, and then hid it somewhere else. So they have this sense of, am I being observed? And if so, maybe hide differently. So that's where ideas about theory of mind in the popular literature, you'll see that kind of being put forward of saying, is it beyond social cognition in a sense of Patrick knows that I hid it here and Patrick's going to want it, so I better wait till Patrick's not here to know that I'm going to move it somewhere else where only I know. And you kind of go, there's a lot of I and other statements in
yeah,
that kind of reasoning of I have a theory that I have a mind and you have a mind and you can know things that are distinct from what I know. And then also tool use. So specifically, I think the Caledonian crows, this is where we observe them doing things with twigs that look pretty sophisticated. And, I once saw a young chimpanzee up in Tanzania, and it had a long stick, and it was at the termite mound,
yeah,
like
yeah.
kind of poking and
Threading,
pulling
yeah,
things out. And you go, well, that was an interesting behavior. And then you watch some of the videos of Caledonian crows, and you go, well, this is very interesting.
it's the same
And
behavior.
it's the same behavior, right?
Yeah,
And how do we make sense of that? And as you said earlier when you were reasoning through the songbird question, well, they don't have the layered cerebral cortex, or so that mammalian neocortex. So instead, we have to ask these questions of, okay, if it's not the mammal version of the neural substrate, what is in their little bird brains that are, I shouldn't even say little, that sounds very pejorative. What is in their bird brains that is underlying these kinds of behaviors that we would describe back to the vehicles conversation in very sophisticated terms. So they have a structure. So the pallium or the NCL is kind of this analog of the prefrontal cortex, back to those kinds of mappings I was talking about. They have pretty dense brains. I think the forebrain, they have a higher density of neurons than we'd even see some mammals, and more kind of comparable to primates when we do these ratios of brain size
yeah.
and what's going on there. So another really interesting case of you're describing behavior, and then there's very different perspectives of kind of what it takes in the substrate to accomplish those behaviors, and what can we say is more directly analogous, and why are we making these comparisons. So corvids, big fan.
Oh yeah,
Big fan.
they are super awesome. There's you cannot say too many good things about them. They I was reading recently, and I'll probably talk about this a little bit more. This book called Ways of Being, that make some bones about corvids, and there's a story in there. They have, for ravens and crows, they have an invocation that they will give to each other when they take off, and the invocation is unique for the individual. So it's almost like they have names for each other.
Yeah.
So they'll start to take off, and they'll turn to their friends, and they'll say grok, and that means grok is the one on the left, and cork is the one on the right, and they don't mix them up. And when they say grok, only grok takes off and goes with them. But if they say cork, only cork takes off and goes with them. And they always get it right, and they always know their names. They always invoke each other in that way. And they like humans too, and sometimes they'll give you one.
Yes.
They'll have it. They'll be like, come with me,
special,
and they'll have a name for you, right? You can get them. People have trained them to do all kinds of stuff. If you can reward them, there are people who will reward them when they bring them money. So like if you bring a bill or a coin back. Yeah. And at first they were like, well, if people drop stuff, they'll clean it up for me, and I'll give them a little peanut or a mealyworm or something. But they will steal. They will start snatching down people's hands. They will find
stuff to bring you.
They're very good
I think
at that.
some parks have trained them to dispose of cigarette butts in a little like crowy treat dispenser kind of thing. And Nikki Clayton in Cambridge, fabulous researcher. She was in the department that I was in, did corvid research. There's videos, BBC has videos with her sort of showing some of the abilities of her birds. And as you say, they don't just like and dislike people, they can hold grudges. So if anyone's ever angered a crow, it will teach other crows to not like you. So be careful with that one. And I think also if you're going to be a research student in Clayton's lab, you had to be a little careful, because if the birds didn't like you, like, sorry, you have to do another PhD project. This is just not going to work out. So there's probably a very tense period when you first get to go out there, get introduced and see, like, is this relationship going to be a working relationship, or have you been rejected from sort of what you thought your graduate program was going to be?
Yeah, they seem to be having one of the experience. This is sort of an interesting place to kind of get into how much do we want to anthropomorphize them?
Yeah.
And up until this point, I think we've been mostly working with nervous systems where they don't really ring human that much. We mentioned it a little bit with jumping spiders. I feel like usually when humans are talking about how smart something is, they are implicitly saying how much like a human is it.
Yeah, absolutely.
And so with the jumping spider, it's like they are very visual. And so we think that they are much smarter than other kinds of spiders, because other kinds of spiders don't usually organize their life that way, right? But jumping spiders organize it around the sense that we also organize their life around, and so we give them a bump, right? Corvids are, oh man, let me run down the list, right? They are visual, right?
Yes.
They are social, right? They talk to each other. They communicate through speech, speech-like productions, right? It's not quite as elaborate as our speech. They can be taught to talk, right? They can use English, right? At about the level of, like, not quite as good as parrots. Parrots have much better pharynxes for this, but they they talk at least as well as Tom Waits does, and they rear their young in pair bonds, right? They are highly social, right? And the other thing that I often throw into this is that they're a weed species. Right, that they're opportunistic omnivores who go into any environment and just try to figure out what the empty niches are and steal a little bit here and there, and that's exactly the list of human traits, right? Yeah. And so I think that we tend to put them in that category. Like, we see ourselves in them,
we do,
in ways that I think are biologically reasonable, right? And so I often think that, like, it's a tough philosophical question to say, like, should we be anthropomorphizing them? Obviously, their lives are very different than ours. We shouldn't just be just seeing ourselves in them. We're not really understanding them. It's just an echo of ourselves. They have a different way of being in the world. It's like, that's true, but also their way of being in the world is a lot more similar than, like, a sea slug's.
Yes.
And so I'm not sure that it's wrong to anthropomorphize them. And I've always felt like with corvids, they seem to be having such a great time.
They seem having a great time. Oh yeah, absolutely. And it is where it's, you're looking, like, on balance, this feels far more similar than different. Then, maybe we don't want to sort of poo-poo some of the anthropomorphizing. It's very common in the popular space, and then also has a popular pushback as well. Of like, you couldn't possibly understand.
Right.
So we've got And we've gone up this progression, and you made a joke, we made a joke together at the beginning around brains being optional. So maybe it's time to talk about humans. What was that about?
Oh, okay. So in addition to kind of looking at variations among brains, neuroanatomical variations among brains that occur kind of naturally, like in a population,
Mhm.
I worked in a neuropsychology lab, which means you look at variations among human brains that are exceptional, typically
Mhm.
due to injury
Yep.
or disease. And there is a very famous patient who is written up in the Lancet, and I'll put a link to this article. And the article has the most wonderfully boring name. It's something like neuroanatomy of a postal worker or a white collar worker
Yes.
or something like that.
White collar worker.
Yeah,
Yeah.
you would never know. And then you see the first picture and you're like, holy, oh my, what is going on?
Yes.
So there was a French postal worker, a French white collar worker
Mhm.
who, worked in the French bureaucracy somewhere, who came in complaining of headaches essentially. And he had
Mhm.
previously, when he was younger, had a shunt put into his brain because he had hydrocephaly, he had pressure on the brain.
Mhm.
And when they fMRI'd him, his ventricles, there are liquid filled spaces in the middle of the brain called ventricles, had expanded to essentially fill his entire skull. And
Yeah.
the amount of brain that is left is a tiny thin shell on the
Mhm.
outside of his skull. It is less than a centimeter of brain thick all around his brain,
Mhm.
tiny weight, just huge volumes of liquid. They were able to give him some relief by putting in a new shunt, but shockingly, he was more or less normal.
Yeah. More or less. Yeah.
Like, it had a slightly below average IQ, but
It's
even that
a
doesn't.
high 80s maybe, but even that, as you said, we're not big fans of IQ
Well, but
anyway.
like,
And if we look at the functional, married, children, job,
yeah,
everyday life, you would interact with this person and not think,
absolutely.
that there's not much there.
And even if you're betting all your chips on that, 1 in 3 people has that IQ. Right? The odds of just finding someone who has 75 IQ are pretty good, right? Like, I would not
Very,
pathologize
very good.
this, right?
No.
And so he's just walking around living his life with virtually nothing in his
Mhm.
skull. And so
Yeah.
apparently, not just different variations among animals are
Mhm.
it is not just that different variations among animals are possible, but
Mhm.
tremendous variation among humans is possible, including up to the point where you can lose huge amounts of your brain and lead a
Mhm.
perfectly normal life.
I think it also just shows us how plastic and robust the systems are. And you and I had a conversation in the past where you said brains are a system that decays gracefully. And I thought that was a really interesting quote. I'd love you to just take 30 seconds to kind of
Yeah.
explain the significance of that idea.
One of the things that you look for there are different ways to judge neural models, really. This is really something that comes out of computational modeling. There are different
Mm-hmm.
ways you can judge a system. One way you can judge it is it fast? Right?
Yeah.
Is it efficacious? Does it solve a lot of different tasks? Is it
Mm-hmm.
powerful? Right? So there are all these ways of evaluating it. One of the ways that is not commonly used in computer science, but is used a
Mm-hmm.
lot by computational neuroscientists, is robustness. And
Yeah.
there is this specific parameter called graceful decay, which
Mm-hmm.
is you say, I'm going to take this system and I'm going to remove processing power from it. Basically, you're deleting
Mm-hmm.
neurons. You're blowing up cells one at a
Yeah.
time. And I require the system to have no more than linear decay. So if I take out 1% of the cells, I want 1% worse performance or better. And there are some systems that have better than that degradation over the short term.
Mm-hmm.
So you can take out, 10% of their processing power and they'll only go down 1%. So you can delete huge amounts. You can, have this shell brain of this French white collar worker,
Yeah.
and they're still working more or less okay.
I have one last one that I just am too attached to not talk about, but I want
Good.
to flip the script because we've been starting about sort of the vehicle hierarchies and the brains that underlie certain behaviors. But I want to start with the brain and have you react to what I'm going to put on the table. So just
Okay.
flip things around differently. So you got a mammal. It's
Okay.
got a big brain, significant EQ, sort of brain body size. It's got an interesting auditory system. Looks like there's a couple pathways. It's more elaborated than you've probably seen in your general mammalian comparison.
Okay.
If we zoom in on the cingulate sulcus, we find this highly elaborate paralimbic lobe with really dense limbic connections. And then the insula is also looking like pretty interesting and more sort of
Okay.
elaborated than you might expect to see. So when confronted with something like that, where does it sit on this hierarchy that we've been making from C. elegans up to humans? Or how do we make sense of something that we haven't really seen in humans, great apes, or some
Okay.
mammals that we typically think of as being highly intelligent?
Okay, I can think. I almost want to start naming animals. I want
Oh,
to
you
start
could
making a
probably
guess.
guess this one.
I have two. There are actually two. I need to.
Hmm.
I can ask a question that will distinguish
Yes.
between them. Okay.
Okay.
Is it bigger than a bread box?
It
Oh,
is.
okay, okay. Then I know what it is. Okay. So I was between two. I was between
Yeah.
chiropterans, bats,
Yes.
and
Yep.
the with the elaborate auditory system,
Exactly.
and cetaceans, the
Yes.
toothed whales. And
Yeah.
so I'm guessing this is a toothed whale.
This is indeed a toothed whale. This is specifically, I was looking at Arsinus orca, or the killer whale, or the orca. Killer whales, fun fact, are not whales. They're in the dolphin family. So Delphinidae, a couple species of dolphins would have exactly the same kind of characteristics that I'm describing. So we are in with the toothed whale. So we see a brain like this. What? How do we make sense of this, or what do we sort of translate to? This is interesting. Here could be some implications.
Well, I mean, you said that we were going to jump off the hierarchy and not go to, like, not
Hmm.
continue to go up, but I think we have. I just think that orcas are
I
better
think we
than
have.
humans. Like, they have a better life. They hang out in the water all day. Like, they swim
Hmm.
around. They're awesome. Yeah, they have some of the. So I would say that they, much like we did with corvids, we can make some of the same arguments. You know, they raise their young, they live in social groups, they're
Yeah.
opportunists, they have a wide range of prey species. They're a little bit more carnivorous, right?
Hmm.
Because their
Yep.
bodies are really expensive to maintain,
Yes.
essentially.
Yeah. Calorically expensive to
Calorically expensive
maintain.
to maintain. They
Yeah.
have elaborate audio communication, right?
Hmm.
They have good vision,
Yeah.
and so oh, and they are a fairly clinally distributed. They're clinally distributed. They're a weed species.
Hmm.
They go all over the place, right?
Yep.
And so in many ways, I would say like yeah, again we kind of have the human measure here, but
Hmm.
now we've got kind of this case sort of like the electric fish where they have this unique sense and they live in such a radically different environment.
Hmm.
You know, corvids fly around, and I'm sure that that dramatically affects their experience, but
Hmm.
they also like are terrestrial. And
Yeah.
underwater is a totally different experience. I assume that the limbic elaborations you're talking about is because they basically don't sleep, or they sleep. They have phased sleep, phasic sleep.
There's some interesting sleep, yeah, phase
Yeah,
sleeps that they do, particularly with the dolphins.
yeah. But that's all. That's what I've got. Tell me more about the elaborations here. I'm dying to know.
Yeah, and I mean, and this is where things get, I think, open to interpretation pretty quickly because, as you say, we're looking at structures in the brain that we haven't seen look like this before, and then we look at their ecology and their behaviors. They say matrilineal pod structures. There's clearly a lot of social cognition taking place. There's interesting communication. They sort of talk about, oh, there's different dialects, or you can tell different pods communicate in slightly different kinds of ways. Dolphins, I was actually, as a high school student, at the conference where they first presented mirror self-recognition in dolphins, and was that wild to see the videos where the dolphins are going up to the mirror and they'd been kind of drawn on with a non-permanent marker, and they were like contorting to look, and oh, then they were kind of like opening their mouths to
Yeah,
try to look, and you're going, well, this is really interesting behavior. And this is where a lot of the popular interpretation, and I'd say this is where the popular interpretation and a lot of the neuroscientists will be on board as well, say, ooh, this looks like, when we think of the limbic regions, the paralimbic lobe, awareness, social cognition, maybe linking memory and learning and emotion could be something that comes into play here. The insula, self awareness, social emotions, maybe tying things to motor control as well. And this is where, as you say, we've kind of feel like we've blown off the top of the
yeah.
hierarchy, are observing something we don't know what the subjective reality or sort of what is it like to be would look like, but we have a clear sense of feeling like we'd be comfortable making the case that there's something here that is significant and different and very
Yeah.
interesting.
I think that for me, oftentimes with these species, especially species like whales
Mm.
that are just like toothed whales that are just tremendously developed and
Yeah.
obviously intelligent in this incredible way, you're almost drawn to like sort of like poetic illusions. Like you
Mm-hmm.
actually have to use creativity in ways
Yes.
that kind of make them characters and make them like like address the differences in their experience in ways that are, kind of narrative and poetic. And there are these, there are some stories, like there's a bay in Japan where there was a pod of orcas, and one of the, I think it was like a boat collision. I think it was an accident, not actually intentional violence, but there was a boat collision with like a baby orca who died out of it. And the orcas have never returned to that bay.
Yeah.
Right? No orcas. Not that pod, no other
orcas.
pods. They've never
No
been
one.
back. And that is like almost chilling. There's there are some experiences. This is in the news much right now, that they've started attacking certain kinds of boats.
Oh, it's fascinating.
And we're not sure
Yes.
why.
Is it? They have fads, so I'm from the Pacific Northwest, and I've had the pleasure of an orca coming right up to my kayak, which was quite both like thrilling and terrifying in
Yeah.
equal measures. I kind of paddled backwards like that would do something as it came right underneath, and all the fish started jumping around the kayak. But there was, I think it was the 80s, the salmon hat fad. And
Yes.
fad is the word that we use for it because we have no other way of explaining this behavior. But one of the orcas, I think it was maybe one of the more like matriarch females, just started wearing a salmon on her head. And there are pictures of this. They're sort of grainy, old newspaper photos. But if you're picturing a whale swimming along with a salmon draped on the top of its head, that's basically what this looked like. But then the behavior spread and it caught on, and other orcas were doing it as well in the region. And they was sort of like this like hot summer fad of wear your salmon hat, and then it stopped. So it was like patterns of behavior kind of rise, spread, and then go away. And in your case, patterns of behavior can certainly arise and spread and persist for a very long time. So maybe it's time for some wrap ups and recommendations, because I think we've gone through a lot of different topics here. And so this is where I failed to come up with just one. And I tried hard, but to be honest, for every brain or organism that I talked about, I would have, okay, if you're interested in orcas, then please go look at this. So I didn't even get to talk about cephalopods, or an octopus would be something that I think is quite interesting.
Yes.
So instead of a media recommendation, I'm just put a little challenge on the table. And that would be, this might be one of those times when large language models would actually shine. So I would say pop open your preferred one, or just our dear friend Google. Think of an animal that you find really interesting. I suspect maybe octopus will prime some people. I bet people that have pets might think about dogs or cats coming to the forefront there. And just put in a very simple prompt along the lines of what is interesting about this animal's brain and behavior. And you'll probably get an initial starting point summary that mimics some of the kinds of points we've talked about over the course of this conversation. But I would say don't just take it at face value, interrogate it, and just take a moment to really reflect on some of the meta questions that have come up of how would we characterize this in a relative sense? Is it appropriate to characterize it in a relative sense? That anthropocentric argument, you'll probably see it bleeding through the way that an LM will talk about the answers to these questions, and questions about how do we perceive them rather than what they are might also be just worth 5 minutes of reflections. This is a sub 10-minute task, which feels fair after we've been giving people books and movies and big things to do. But I'd encourage you to do it and just pick something of interest and work through it for yourself and see what you find.
Actually, that has reminded me of one, which,
yes,
if you're interested in the intelligence that can arise from relatively simple nervous systems, one of the best works on this is actually Charles Darwin's last book, The Importance of Earthworms. Darwin's one of the few scientists that I actually think kind of lives up to the legend, and not because he's like really like smart like Newton was, but because he's like genuinely like kind and down to earth and just a sweet person who I actually like a lot. And in his older years, when he couldn't do much science outside of his garden, he did an extensive study of earthworms, where he would study them in sort of laboratory conditions, and then he would study them in more naturalistic conditions in their actual environments. And he would say they are very dumb in laboratory conditions, right? Pretty much the only thing he could describe in laboratory conditions that characterized their intelligence is that they did not like lemon juice. But then he gives all these amazing examples of the intelligence and the cleverness of how they do stuff, that they could build. They would take pine needles and use them to reinforce their burrows. but then they would
Mm-hmm.
make sure to tuck in the pointy ends so that they wouldn't get stuck on the pine needles. And that's fairly sophisticated.
Yeah.
And so I really like that book. It's not a long read. It's
Mm-hmm.
historically dense. It has old
Mm-hmm.
language in it, but it's a very sweet sort of citizen science, old age
Mm-hmm.
citizen science from one of the greatest scientists who ever lived. So
And I think that's a really helpful note to end on because I think it also would extend what I've been suggesting too, and that's taking the time to think about what does naturalistic behavior look like for these organisms. So take them out of the lab and really think through on their own terms what behaviors are demanded of them to exist, interact, be successful, procreate, and that can lead to really interesting questions of rather than teaching the crows and the dolphins to talk, let's try to say, well, what does language actually look like for them? And there's some very interesting insights to be had if you go down that garden path rather than the former. On the note of scientists we admire, I think we're going to be looking ahead to engaging with the works of the great Oliver Sacks. I
oh
think
yeah.
most neuroscience students will absolutely encounter at some point, and we definitely want to take some time to dig into some of the interesting examples from the man who mistook his wife for a hat. So we look forward to this conversation in the future, and we'll see you next time.